Question 1124 is about telomerase, and the question is, essentially, let me read it here. In 1994, telomerase activity was discovered in human cancer cell lines. Although telomerase is not active in human somatic tissue, this discovery indicated that humans do contain the genes for telomerase proteins and telomerase RNA. So those would be the TERT and the TERK components. So since inappropriate activation of telomerase may contribute to cancer, why do you think the genes coding for this enzyme have been maintained in the human genome throughout evolution? And essentially it's because the telomeres will shrink. You know, if we don't have telomerase expressed in some cells. So the next part of the question is, are there any types of human body cells where telomerase activation would be advantageous or even necessary? So let's see if we can, watch the telomeres shrink. If we can diagram the telomeres and watch them shrink as the DNA goes through various replication cycles. And let's just look at one telomere. I don't know if this is going to work out correctly, but sometimes it's hard to diagram this, and you have to go through several cycles to see it. But let's give it a try. Okay, so we're just going to look at the end of one DNA molecule. So this DNA molecule is, extending that way. Let me see. Okay. So we're looking at the right end of this DNA molecule. And we're not going to worry about the overhang or anything like that. That is part of the T-loop, D-loop model of telomere protection. We won't worry about that. We're just replicating the telomeres here. Let's make this the 3' end and this one the 5' end. Okay, so during replication, these are going to come apart. And we're going to have to replicate both of these semi-conservatively. And it might be quicker for me not to draw these little nucleotides on here. Okay, so we're going to need an RNA primer right here. And then we can synthesize or replicate this strand, copy this template here. And this is the 3' end, and this is the 5' end. And this one right here, okay, this is no problem. Since this is the 5' end, then this is going to be replicated all the way up to the end here. Okay, so now we have two DNA molecules. Well, we haven't finished yet, right? So we still have this RNA primer in here. So eventually that RNA is going to be lost, but it can't be cut out by the DNA polymerase I equivalent in humans, right? Because there's no primer over here. There's no primer over here for DNA polymerase to start polymerizing DNA and removing and at the same time removing these RNA nucleotides, these ribonucleotides. So what I'm going to do here is just erase these. So they're going to be lost. Some exonuclease is going to cut them out, or they're just going to, I don't exactly know how they're, or what the thought is on how these are lost, but they are lost. And so eventually we end up with a DNA molecule that looks sort of like that. Okay, so we got two DNA molecules here, and now okay, let's say these are both in different cells, and now let's see. Let's look at replication of, let's see, this one right here. Here's my pen. It's not working. Okay. So, okay, this DNA molecule is in a cell, and this is in another cell. And let's watch. Okay, this cell is going to divide through mitosis, and the DNA is going to be replicated first. So, okay, DNA molecules are going to separate. Okay, so 3' end here, 5' end here. So this one up here is I'm drawing up at the top, and this smaller one down here I'm drawing down at the bottom. Okay, now here's the 5' end here, 3' end here, and again I'm going to use a different color. Let's use blue. Oh, for the RNA primer, let's use blue. So we're going to need an RNA primer at the end here, and the RNA primer for this one is somewhere, further up on the DNA. So DNA polymerase 3, or the equivalent in humans, is going to replicate this all the way to the end, no problem. And this one is going to be replicated, just right here as Okazaki fragments. And okay, we have the same problem. So this is going to be lost. Okay, so we have 2 new cells here. I wonder if I can, no. Can I draw a circle around these? Nah, that's not very useful. So 2 new cells here, and I'll use this white pen. So one of the cells gets this DNA, one of the cells gets this DNA right here. Okay, so we can see that okay, we started here with a telomere of this length. After one round of replication, well, we ended up with a telomere of this length right here, the same length in one cell, and we ended up with a slightly, stranger looking telomere in the other cell, slightly shorter on one strand. Now when this cell replicated, it gave rise to 2 cells, one that looked identical telomere, and another one with, slightly, telomere was essentially the same length of this red piece right here, but we lost that little overhang. Okay, so now we have a cell with, as a telomere that is distinctively shorter. It's missing this piece up here that was found in the original guy up there. So if you continue this process, and you follow these telomeres, you will see that in a large population of cells, you have the telomere shrinking. Now, eventually, when in some of these cells, you're going to have, at least this is the hypothesis here, this is a long end of the chromosome, and let's say that those telomere repeats start like somewhere around here. So this is the telomere region, where we have all the telomere repeats, and it forms that T-loop, D-loop. At least one of the models say it forms that T-loop, D-loop to protect the telomeres. And then over here we start seeing genes. I've got a gene here, and so on and so forth. So if these telomeres are shrinking with each round of replication, there are many rounds of replication, eventually we're going to end up in here. Then we're going to have a cell with no telomeres, the ends aren't going to be protected, and eventually we're going to lose these genes, and the genome of that cell is going to be really unstable, and eventually the cell is going to be lost. The exact mechanism it is lost, it could be lost by programmed cell death, or it could just be dysfunctional. It starts losing genes. For whatever the mechanism, the loss of the telomere through progressive rounds of replication is thought to lead to cell death and eventually over time thought to be a major cause of the aging process. At least that's what a lot of the evidence that we see, some of the evidence is showing. So now telomerase will actually extend the length of the telomeres. So it counters telomere shrinking through each replication. So the telomerase enzyme with the reverse transcriptase in the RNA component will actually add repeating subunits of invertebrates. This would be TTAGGG, adds those repeats to the ends of the telomere, so it keeps them from shrinking. But it doesn't do that in all cells. Telomerase is not expressed in all cells. You know, what the problem said, telomerase expression was found for the first time in humans in cancer cells. So it turns out most cancer cells, most tumors, express telomerase. And human somatic cells are not supposed to express telomerase. And the thought is it's a defense against cancer. So if a cancer cell, if cells start to become cancerous and they start to replicate uncontrollably, maybe loss of the telomeres will cause those cells to burn out. And prevent them from developing into full-blown cancer. But there are cells in our body which require telomerase, and those would be germ cells and stem cells. So germ cells and stem cells, and I don't know a whole lot about stem cells, but these are cells which we need to replicate continuously throughout our lives, and they do express telomerase. At least the thought is they do express telomerase, and it keeps the telomeres from shrinking and keeps our germ cells stable. And our stem cells able to keep producing new cells. So telomerase is expressed in a select number of human cell lines, human tissues, and but it's not expressed in most human somatic cells.